Hi, I'm Mark Hagan, Regional Account Manager with BrettYoung. Today I have with me Glen Hawkins, the agronomist with DL Seeds. DL is the primary genetic supplier for BrettYoung's canola line. Here we are today in the Spruce Grove Clubroot Nursery to talk about first generation and next generation Clubroot products. Products like our 6076 have a great fit on your farm and maybe the answer to some of your issues. So Glen, what are some of the key areas of concern when it comes to breeding for clubroot potential?
Well, Mark, it comes down to what type of clubroot resistance you're looking for. Like you said, this is our next generation clubroot genetics. So this is predominantly our 3A nursery. We do carry genetic resistance to 3H as well, which would be considered first generation resistance, and that would have been the first generation products that farmers first saw when clubroot began to spread throughout the Canadian prairies.
So can you explain in more detail what the difference is between a first generation and a next generation clubroot?
Sure. Well, the first generation was basically a gene derived from Mendel. Mendel was a European variety that had resistance to pathotype 3H, which was the dominant first generation pathotype here in the prairies. We then took Mendel and incorporated it into our breeding lines. Unfortunately, after a number of years, that resistance began to break down. And what we see in this nursery now is resistance to one of the newer pathotypes on the block, 3A, which we term second generation resistance. And we're breeding lines like 6076 and incorporating those new genes into our portfolio to provide robust resistance against this pathotype.
So what makes a variety like 6076 a next generation clubroot variety?
Well, we basically touched on that part of it. Basically, it's the fact that we have Mendel, which provides our first generation resistance, or 3H, in our background. And we've added a second set of genetics, and it gives us resistance to 3A, which is the pathotype in this nursery, which is termed next generation resistance to clubroot.
So how is molecular genetics important in determining clubroot race?
Well, as we learn more and more about clubroot, we begin to realize that it's not a one gene fits one pathotype model. We need to have several genes or clusters of genes with helper genes. Now, if we can get markers, molecular markers to those bits and pieces, the breeding and selection process can be accelerated. So from the time we identify the need of a new pathotype or susceptibility, the time it takes to gain resistance, and if we have markers, is hopefully less. So we can use bigger populations, use molecular markers to screen those populations for resistance, and get them in combinations in the field faster.
So being that we're in a clubroot nursery field today, what types of things are you looking for in these fields to help in the next generation and pushing clubroot resistant management further?
Well, if we're looking at selection in our field here, the biggest thing we look for is uniformity, right? If we have patchy clubroot, we get patchy results. Fortunately, this field here has great broad levels of pathogens, so we get nice uniform field. If I was to talk to a farmer though and talk to him about his clubroot issues, the first thing that comes to mind is genetics. That's what we provide. But the second thing is just a strong farm management. Great rotation, great weed control, and again, great rotation, right? On a solid 4-year rotation, you can minimize your risk of total wreck on clubroot and the buildup of new pathotypes if you're using a resistant one already.
So Glen, you touched on this a little bit already, but can the Western Canadian Clubroot Challenge be solved purely by genetics, or are there some other strategies such as rotation that a grower would have to take into consideration to manage it?
I think it's right. It's a combination of both, right? Genetics is going to play a huge part, but mitigation and rotation and keeping that clubroot load to a minimum on your farm is probably one of the most important aspects of clubroot management, right? If you don't let that clubroot load get to 10 to the 6, 10 to the 8 spores per mill of soil, it's a more easily manageable disease, right? Your spread is decreased, and your ability or the inability of, or the potential for a wreck, which we call a wreck, where your whole field is lost to clubroot because you're growing a susceptible variety, is also decreased. We know that over time, Clubroot spores decrease rapidly in the first 2 or 3 years that they're left in the field without a host. So good rotation, strong clubroot genetics, and in those off years, weed management. We walked through the field today and we found weedy relatives of brassicas that are also susceptible to clubroot. So you want to make sure you're doing strong farm practices, good weed control, rotation, and strong genetics.
Thanks, Glenn, so much for your time today and going through some of this information on first and next generation clubroot with us. It certainly helps to give growers a better understanding of the nature of the beast we're dealing with and how we can best manage it. If you have any more questions, please contact myself or your local BrettYoung representative, or visit our website at brettyoung.ca for more answers.